Hello, and thank you for joining to this year's research update. My name is Silvia Pradas, and I am the DRPLA Research Manager for Cure DRPLA and Ataxia UK. So in this year's talk, I'm going to go over who we are and what we do, and then I'm going to focus on some of our projects. That includes the N-of-1 clinical trials that are happening in the US. I will also focus on a selected number of projects funded by Cure the RPLA, and then I will also explain some other projects and relevant news that are of interest to this community. And finally, I will close by explaining some opportunities to take part in research studies. So Paul and Andrea Compton, they have a son that was diagnosed with the RPLA in 2018, and they decided to create Cure the RPLA, a non-profit organization to connect families, physicians, and scientific investigators to further the RPLA research and work towards a treatment. Cure the RPLA has a partnership with Ataxia UK, and we've been working together for almost 6 years now. And in this picture, you can see who is part of this team. So up to now, we know that DRPLA is a heterogeneous condition, is not fully understood, and there can be significant differences between individuals. Adult onset DRPLA typically it's characterized by the first symptom appearing after the 20 years of age, and in this case, most people with adult onset DRPLA, they tend to have less than 65 CAG repeats, and it's characterized by loss of coordination, involuntary movements, dementia, and behavioral changes. On the other hand, juvenile onset DRPLA is when the first symptom appears before the 20 years of age, and typically in this case, people tend to have more than 65 CAG repeats. It's characterized by seizures, ataxia, sudden muscle jerks, and progressive intellectual decline. And we know as well that there are no approved treatments that are specific for the RPLA. And because of that, the clinical care focuses on managing symptoms individually. So I'm going to start by talking about the N-of-1 trials. These are trials that involve a single person, one therapy for a period of time. And what they do is that they collect clinical data and biosamples before and during the trial to determine if the therapy makes a difference. When we talk about N-of-1 clinical trials, I really want to emphasize how different these are. So to do an N-of-1 clinical trial with one person, the time it takes to go from the lab to the clinic is much shorter because they require less experiments to be done at the preclinical stage, so before it's tested first in humans. So N-of-1 clinical trials, what they allow scientists to reduce the time it takes to develop a drug. But obviously that also means that because they don't do as many studies as they would do for other types of clinical trials where they tested in hundreds and thousands of people, what this means as well is that at the end of the N-of-1 clinical trials, at the moment, there is no path for this drug to be approved and reimbursed. So that means that for now, N-of-1 clinical trials are experimental. So how are N-of-1 clinical trials possible in the US? They are possible because there are two elements that coexist, and these are crucial for this type of trials to happen. First of all, in the US, there is the Food and Drug Administration, the FDA, that has created a regulatory path to test new drugs for rare diseases that affect a very small number of individuals. So we first have a regulatory environment that supports this type of trials, and then hand in hand we also have the N-Loren Foundation that charitably provides experimental antisense oligonucleotides to treat people with nano-rare conditions that affect less than 30 people in the US. We also know that DRPLA has a low prevalence in the US. There's only 11 people, according to the patient registry, with DRPLA that we know of. And also, at the beginning, when NLOREM was created, Cure DRPLA worked with the NLOREM Foundation and Dr. Jennifer Bain at Columbia University to explore N-of-1 clinical N-of-1 trials in the US using antisense oligonucleotides for the DRPLA. So, what are ASOs and how do they work? ASOs are short for antisense oligonucleotides, and these can block or silence the harmful message that causes the DRPLA. If ASOs were to be proven safe and effective, they could become a treatment option for the DRPLA. And what we know is that in the DRPLA there is the ATN1 gene that is expanded. So there's a part of the gene that has more copies than what it should be. And this is what we call the CAG repeat. So if there are more than 48, this is considered a clinical diagnosis of the DRPLA. And we know that when the CAG repeats expand more than 48 times, they cause this protein to be longer than usual. Therefore, cells struggle to cope with this extended protein, and what happens is that it ends up being accumulated in cells, and this creates toxicity. What ASOs can do is that they can block this step from the gene to making a protein, and they therefore can potentially reduce this abnormal accumulation of this protein that is extended. So now I'm just going to focus on the N-of-1 trials that have happened to date. The first participant, before starting on the trial, presented with ataxia, dystonia, epilepsy, intellectual decline. He also struggled to speak and was unable to walk more than 50 feet without a wheelchair and needed full support for daily activities like dressing and feeding. Up to now, this participant has received 8 doses of this antisense oligonucleotide through lumbar puncture, and he started in February 2024. It's really important to highlight that today there's been no ASO-related adverse events or no serious adverse events, and that's important in terms of safety. The clinical benefit has been reported in some domains. So, for example, they've seen an improvement in a speech, a reduction in the number of seizures, and also days without seizures. Swallowing has improved as well, and so has the bladder function. They've seen a stabilization in coordination in the arms and legs, but they've seen a worsening in the balance while sitting and standing, and this participant is no longer able to walk. And also caregivers have seen improvement in engagement and perhaps some personality changes. So for the second participant, before starting on the trial, had been cared for in bed for 20 years, needed full support for daily activities, was nonverbal, fed through a gastric tube, and also had a tracheostomy. Up to now, has received 5 doses of this ASO since November 2024. And again, very important to highlight that today there's been no ASO-related adverse events or serious adverse events. They've seen subtle changes that have been reported by the participant's mother. She has said that the participant is more alert. She's able to track people and items with her eyes, and she has more enhanced facial expressions. So the third participant, the FDA has agreed for this participant to receive the ASO, and the treating clinician has obtained all the paperwork that is needed for this to happen. So now they are just waiting for the participant to actually start the treatment. And there's also been a fourth participant that has that has submitted a case to the FDA, but they still need to work through all the paperwork. So for this participant, the treatment has not been started yet. So it's really important, again, just once more to highlight that N-of-1 trials are focused on safety. Data from single trials can be really, really difficult to interpret. It's difficult to understand what can be explained by the ASO treatment, what can be explained by the progression of the condition, and these changes from individual to individual. And obviously, the evaluation of these ASO effects is ongoing. As you've seen, only 2 people have received this ASO so far. There will be potentially 2 more people receiving it in the US, but obviously it's still very early days. So now let's focus on expanding beyond the US. As I was saying, only the FDA in the US has these specific guidelines, but no other countries have such specific guidance or established regulatory path for individualized treatments. So what we know is what is required to pursue such trials outside the US. We know that obviously we need to have people with the condition that they need to find a physician at a place that supports these type of trials. We also need to have a physician or a clinician that understands what these trials are about and are supportive of these type of trials. However, a lot of physicians really struggle to get institutional support, or they don't have the knowledge of the regulatory pathway. Most times is because such regulatory pathway does not exist. And also physicians need to secure funding in order to have conversations with their institutions about these type of trials, what are the risks, what are the liabilities at the institution side. And obviously, because these are not approved treatments, these remain experimental, there's funding needed to administer these ASOs. From an institution perspective, most of the times they don't understand what N-of-1 trials are because it's not been done before. So the type of conversations that need to happen about contracts and all this paperwork, again, they lack the expertise. So a lot of the time, from a physician and institution perspective, it's difficult to even find who to talk with because there's no one with the specific expertise for this. Obviously, we can leverage what exists at existing clinicians that are interested in exploring N-of-1 trials. They can have conversations with their institutions about these type of trials, and that's happening already. But again, everything needs to be started from scratch because this pathway does not exist outside of the US. Obviously, Cure DRPLA and Ataxia UK are determined to help more people with DRPLA and their families assess whether N-of-1 trials are right for them. We are looking for UK-based institutions with the infrastructure to conduct N-of-1 trials, and we are also exploring ways to address this lack of funding to administer the experimental ASOs. Obviously, we are also in touch with NLOREM. We are working diligently to help patients outside the US, despite the limited resources from all the parties involved. And we are also in touch with other groups that are in very similar positions. Like, we are trying to share as much knowledge as possible. Like, there are other groups that are trying to set up N-of-1 trials outside the US, and we are all talking and collaborating because sharing knowledge is gonna really help us move the needle forward. So What we know as well, just as background, is that the contract negotiations between NLRM and the US institutions have taken anywhere between 3 months to 2 years. And the biggest hurdle here is funding, um, is the ability to find funding to support these clinical trials. So now I'm just going to move on to a different topic. I'm going to start talking about some of the research projects that Cure DRPLA has funded. So obviously we as an organization are very interested in ASOs, and we also funded projects to test other ASOs beyond this one that is being tested in N-of-1 clinical trials. So we started funding Professor Paola Giunti's lab to do some preliminary work in cells. And then we also funded Dr. Jeff Carroll at the University of Washington. And what these scientists have done is that they've utilized this ASO developed by Bico Therapeutics that is currently in clinical trials for spinocerebellar ataxias types 1 and 3 and Huntington's disease. And this ASO from Bico Therapeutics could potentially also help conditions like the DRPLA. So this ASO has been tested in the ATN1 mouse model that we developed a couple of years ago. And the objective was to test if this ASO could be beneficial in a humanized mouse model of the DRPLA that presents a very severe form of the DRPLA because it has a very long track of CAG repeats. We also explored other types of drugs beyond ASOs. So again, we funded the same researchers to look at omaveloxolone, which is an FDA-approved treatment for Friedreich's ataxia. This is a pill that's taken orally and at a cellular level can protect, reduce stress, and improve energy. So again, we wanted to test omaveloxolone to see if it would be beneficial in a humanized mouse model of the DRPLA with 112 CAG repeats. So the outcomes of these two projects, the scientists collected behavioral tests that showed that the mice treated with the Bico Therapeutics ASO or omaveloxolone showed no improvement when they compared it to untreated mice. They also conducted a large number of neurological exams to quantify the impact of these two treatments, but they did not observe an improve with any of them. There was no evidence that omaveloxolone generated the expected changes in the mouse model. So as for next steps, obviously, the researchers are still evaluating these results and reviewing the design of the study to see if anything could be improved to determine if this didn't work due to the way the experiments were planned or if this doesn't work because these treatments might not be helpful for the DRPLA, at least in the conditions tested, right? Like we always say this was tested in cell lines and animal models, and in the conditions that it was tested, they did not see an improvement. But obviously, there's still a lot of data that needs to be processed. And again, review if there's any improvements that could be done in the lab to continue studying this. What we also believe is that the DRPLA mouse model that it was tested in, as I was saying, they have like a very severe form of DRPLA. So perhaps because these mice are too sick, they did not observe a rescue with the Bico ASO or omaveloxolone. We are also studying new mechanisms. So this is a project that we are approaching from different perspectives. We are funding Dr. Jeff Carroll at the University of Washington, Professor Darren Moncton at the University of Washington, and this is also a really big part of the DRPLA Natural History and Biomarkers Study that is recruiting participants right now. So we know that somatic expansion is a biological process where the CAG repeats can expand in different cells of the body over time. And what happens is that when these CAG repeat expansions increase in number, they may contribute to when and how the symptoms developed. And pharmaceutical companies and biotechs are developing drugs to rescue somatic instability for conditions like Huntington's disease. So, knowing this, obviously, we want to explore to which extent somatic instability is present in the RPLA and it's making things worse. And if that's the case, explore some of these somatic instability drugs because, again, they could also be helpful for the RPLA. So, again, changing topics, I'm now going to focus on external projects. So, these are projects that are not funded by Cure the RPLA and relevant news. So we are really pleased to say that Dr. Melissa Barker-Halinsky was awarded funding from the American Epilepsy Society, and this is a project that's been taken place in collaboration with Dr. Jeff Carroll and Dr. Joanna Koreka. And this research project is to better understand epilepsy in the RPLA cell lines and mouse models. And it's really good to see that other organizations beyond Cure the RPLA are funding research into the RPLA. We are also really pleased to announce that there are 2 Japanese pharmaceutical companies that as of April this year, they announced that they are working together to develop an ASO for the RPLA in Japan. And these 2 companies, again, they've also said publicly that they will seek partners to develop this ASO outside Japan. And obviously, we are going to report updates about this ASO as they become available. Lastly, we also want to share that there is a clinician in China. His name is Shirui Gan, that has published the largest characterization of clinical findings in a group of people with the RPLA. A few months ago, they published a paper in which they enrolled 116 people living with the RPLA across China. 96 of them had symptoms, and 20 of them had been diagnosed, but they didn't have major symptoms yet. And again, it's really great to see that more clinicians are working on characterizing the RPLA and conducting studies. We have a great working relationship with Shirui Gan, and we are trying to set up collaborations. Again, the more data that we can collect on the RPLA, the more it's going to help us advance the knowledge and therefore potential treatments for this condition. And now the last part I really wanted to cover in this talk is taking part in research studies. So why does this matter? So when people take part in research studies, they can help develop treatments now, but also for future generations. They can shape the development of therapies to better meet the needs and preferences of those living with the RPLA, contribute to advancing the RPLA research, and sometimes it also provides this sense of empowerment because you are helping the community directly. It also helps gather information from the people that are the most knowledgeable about the condition. And it also, in some cases, it can also give you access to expert medical care that is not otherwise available. So let's now just review how you can take part in research studies. The first one I want to highlight is the Natural History Study and Biomarkers. So this study is going to provide a description of the RPLA with an understanding of the full range of symptoms and how these progress over time. For this, we or the clinicians involved in this study, they selected a number of assessments that would help capture such information. And obviously, this study is also going to inform the design of clinical trials that may take place in the future. This is a 3-year study, so participants undergo 3 study visits once a year, and they are enrolling people with DRPLA but also people without. And this is an international study. There are many, many countries and sites participating in the study. So this study was set to recruit 157 participants in total, and to date they have recruited 135. So it's really encouraging to see that they've recruited, they've achieved an 86% of the recruitment goals. And as you can see on this table, they are recruiting adults and children, and as I said, they are recruiting people with DRPLA, so these would be the carriers, but also people without DRPLA, and these would be the control participants. So participation in this project, you can really determine how much or how little you are willing or able to do. But just as a background, clinicians collect information on demographics and clinical history, epilepsy and frequency of seizures. They also do physical examinations and functional tests, evaluate speech and swallowing, cognitive assessments, brain scanning, and they also collect biofluids. And again, this study is going to encourage companies and researchers to work on drug development for the DRPLA. The last study is the Cure DRPLA Global Patient Registry. So this is an online collection of questions or surveys. The first time you complete it, it takes less than 30 minutes, and then we ask participants to redo it on a yearly basis because we want to be able to track the progression. And when you do it every year, then it's quite quick to complete and it takes less than 15 minutes. Again, this registry is going to help us characterize the DRPLA, better understand prevalence across the world as well. For example, like when I was referring to the N-of-1 clinical trials, this registry was really helpful because we were able to tell the regulators how many people had DRPLA in the US. So again, these tools are really being used to advance research, but also to advance treatments and help organizations that are developing treatments. This registry will also provide data, anonymized and grouped data to scientists, and it can also help participants or connect participants with opportunities to take part in research. It's been approved by an ethics committee and it's available in 6 different languages. So to date, we have 65 participants on this registry. 54 have symptoms and 30 of them have juvenile onset DRPLA. So that means that the first symptom they experienced was before they were 20 years of age. And then we have 24 adult onset DRPLA participants. There's 5 asymptomatic participants, so that means that they don't have any symptoms that they can attribute to the DRPLA. And then we also have 6 unknown participants. So these are participants that created an account, but they didn't fill in enough questions for us to understand what type of DRPLA they present with. And just to let you know as well that we are working with a statistician and we are planning to publish the description of the participants that are on the database next year. So again, like what is the first most common symptom, what does trajectory look like, and so on. So just to wrap up, obviously, while I presented all these updates, there's a lot of people working behind the scenes to make all of this happen. We really want to thank all the families and all the people with the DRPLA that have participated in studies or have engaged with us for your support, because none of this would be taking place without any of you, and also all the scientists. I was saying, like, this is just a brief update, but there's like so many people working on all these projects, and we have really brilliant minds that have received funding from Cure the RPLA, and we are really, really pleased to see all the advancements in the field. So with that, I'm just gonna conclude. If you have any questions, you can contact Cure the RPLA directly, and we will get back to you by email, and we can also schedule a phone call if that's preferred. So thank you so much for your attention, and I look forward to hearing from you.